Wow, you make me sound good. I am, I am very impressed. So I have to say first, thank you, Terry, and thank you, organizers, and thank you, everyone, for coming out here. This meeting has really evolved. It's just transformed actually into something. And it was just, I was just noticing on a Slack channel, somebody saying, ooh, this is looking good. I wasn't going to go. I'm not able to go this year, but it looks like people are really paying attention to this year in particular. Okay, I do want to also add my land acknowledgement. I respectfully acknowledge the unceded traditional territories of the Coast Salish people, including the Tsleil-Waututh, Coquitlam, Squamish, and Musqueam nations, upon which I live and work and play in Vancouver. It's a beautiful area. I am outdoors every day, enjoying this lands. I also want to acknowledge the beautiful traditional lands and waters of the Kanyankehaka or Mohawk, where we are gathered here. This is just a picture of in BC, and this is a picture here of a park that's in Montreal that I was just hunting around. I'd love to go there. Anybody interested? I am very keen to check it out. Okay, so I'm having an issue with slides moving forward. Just a minute. Nope. Seems to be frozen. There we go. I will just do that. Okay, thank you. I also want to acknowledge I've been really transformed in my journey through science, getting interested in open data and open science, and then getting interested in consulting with different people and doing sort of interdisciplinary sort of investigations. And one of the things I've been transformed with is actually just talking with some Indigenous communities and learning about some of the ways they're doing things and how beneficial some of that work is. And I've learned a lot, and I'd like to acknowledge in particular Mathea Latini, Robert Spence, and the CEDAR research group in Yukon, Manitoba, and at UBC. They are just phenomenal groups that have really, some of the things I'm going to be bringing up really comes from them. So yes, one of the things Indigenous populations do is they talk about introducing yourself, and the point is supposed to be so that you understand their background and where they're coming from as they start to bring up something. So you've already, Terry brought up some of my intros. So yes, I was born in Australia, and then my parents decided to move to Canada to be closer to Scotland, to be closer to home. So I grew up a lot outdoors, in and on water in Ontario, and in trips to Scotland, and a lot of hiking. And through that, I really got interested in life. And like many of you probably here are basically interested in life and understanding life and coming up with things that involve learning about life. But also, like many of you guys here, I'm also a computer geek. I got my first computer, VIC-20, back in the 80s. I just am going to mention this. You're probably going, why am I talking about this? There's actually a point. First, I just have to geek out a little bit and just say there was 3,583 bytes, not kilobytes, but bytes on this machine, okay, of memory available for coding after you, for coding and variables. And there was no hard drive, no floppy disks. There was a cassette tape-based data set where you had to play and record to record my program. And I created my first little game in BASIC of a little boat going around. Of course, you tried to do it as efficiently as possible. And I undergrad did Fortran and Pascal. So I want you to realize, so that was, okay, granted, that was like about 40 years ago, but think about how far we've come in 40 years, and what is probably going to be a future, and how much things could get transformed in the next 40 years. I don't have to say, well, I'll just say it right away. Generative AI, is certainly going to have an impact on us, and I will talk more about that and with some cautionary notes. I also just have to comment that I was a computer gamer, and my parenting partner, as we call each other, Ryan Brinkman, who's in the audience, you've got to check him out. He's a very sort of forward-thinking person. an early adopter, I call him. And but he was always, you know, finding some cool game to play, and we would compete. And I realized I had to read Sun Tzu's The Art of War to figure out how to better combat him. And there is a point to this again, that Sun Tzu's The Art of War, I started to read it in the context of pathogens and how to combat pathogens. And I realized, oh, wow, you know, this is actually really relevant. So my first main point I want to make in this talk is I'm talking about sustainable science that's being sensitive, but I do want to mention the strength and the innovation possible for interdisciplinary integration. And so just this idea of taking pathogen research and putting Sun Tzu's The Art of War and overlaying on that, you know, what I realized was, okay, here's a couple of quotes that are obviously translated, but what is of supreme importance in war is to attack the enemy's strategy. I think of strategy as their virulence factors or disease-causing genes. Next best is to disrupt his alliances by diplomacy. The next best is to attack his army. And the worst policy is to attack cities. And I'm going, holy crap, because that's what we're doing when we give antibiotics. When we give an antibiotic, you are just nuking everything in there, the good and bad bacteria, as we quite call them. And so not surprisingly, resistance movements blossom in those situations. So Sun Tzu was already telling us antibiotics not a good idea. But that is actually what, as Terry was already bringing up, this is what really got me interested in maybe we should be targeting virulence factors more, the true cause of disease. Let's disarm them. I'm sort of like a peace-loving kind of microbiologist, microbial bioinformaticist. And so let's allow the pathogen to survive. Let's not actually kill it. Let's just try to disarm it. Let's try to attack those virulence. So that's what we're doing. And with Amy Lee, and fantastic assistant professor at SFU, is up and coming bioinformaticist. We're working together to look at pathogen-associated genes that are only found in pathogens and not in non-pathogens, learning about new virulence factors actually as we're at it, but finding genes that we can target with drugs that don't actually kill the pathogen but disrupt them and allow the microbiome, all the good bacteria, to survive. So this is just an example of innovation through interdisciplinarity, and there's a QR code. You're welcome to get exercise from lifting your phone up to do QR codes, but I'm happy to share the slides later, but that's just a paper that we wrote just as about the start to this journey. Okay, so how far we've come, though, I just have to say one more thing about that, and that is in the 90s, we had a GenBank versions that arrived via snail mail on floppy disk. I was so excited every time that floppy disk came in because I was like, what new sequences do we have this time? And that I don't actually have to type them in from a paper. That's what we used to have to do, you know? So this is how far we've come, okay? Even since the 90s. And so the first major sequence analysis I did was a phylogenetic analysis, this carbophosphate synthetase gene. It's a gene that takes CO2, ATP, and ammonia and makes a molecule. It's a pretty cool one. It impacts the carbon cycle, nitrogen cycle. And what I realized in there's 2 genes to make this enzyme. And in one of the genes has an internal duplication that is present in all life, in bacteria, archaea, and eukarya. And I realized, oh, wow, we could root the tree of life with this, because this duplication is in all of in all of these, all of life, then that basically occurred before the first cell formed. We could basically just line up all of the, say, the second duplication and root it with the first duplication and vice versa. So this is like the first duplication, second duplication, and it did seem to work pretty well. I mean, all the bacteria grouped together, the eukarya grouped together, the archaea, we had this one archaea sequence at the time, and we were just curious about where it went. And so it grouped with archaea, which is in fitting with what generally we think is most likely the order of how organisms did occur, that there is a bit of an archaeal eukarya linkage. But I have to say, one of the messages I have as a cautionary tale is I got interested in other stuff, and this is one of the problems, is that I would have loved to have followed up with that, but funding directions dictate that you end up going in certain ways. And I Ended up not following this further, but I did read a subsequent paper or hear about a subsequent paper that said, oh, we added more archaeal genes and they were all over the place. Doesn't seem to be very good. I only just discovered when preparing for this talk that actually that paper, I hadn't actually looked at the details of it. They were actually sticking all the genes together. They weren't looking at the duplications in the exact way I was. So I actually really have to redo that analysis with the right sequences. And it just shows you that you have to be careful with the details. It just sticking a bunch of carbon-phosphate synthetase genes isn't going to work because those genes themselves, there's multiple genes and there's horizontal gene transfer, etc. This is a link to that paper. This is just showing the nice symmetry of that molecule, and the green and blue are these two kind of domains that have occurred earlier in life. They're kinases. But the reason I'm giving this example actually is to note the scanning errors that is occurring and the impact this could have. So this is a paper that I went back to that I noticed when I went to the figure. It's only available in PDF, this paper, and it has some decent data, but look at this Plasmodium falciparum, P falciparum. It's like a line with a question mark, falciparum, and this is supposed to be sulfatericus. This is supposed to be S. sulfolobus, and it's a mess, okay? And this is the text that has now gotten into our internet. This is the stuff that AI is going to look at, and it's garbage. I'm wondering how many other papers that were scanned that actually have really good, meaningful data from that era that are now getting literally lost through scanning errors. So I do think there are some implications for AI training of this. This is for phylogenesis. You'll notice there's supposed to be a line here of the number of substitutions per site, and there's nothing because it didn't show up in the scan. Okay, the other thing I started to learn at the time was this idea of this need for standards and organization. And I do want to do a shout out to Will Sow and Emmett Griffiths, who have been researchers, have done some really seminal work in this area. But really what happened was I realized this gene had a million different names and it was a big mess. And in fact, the most frequently used name for this is not actually the one that's designated. I don't know why. This name is used like maybe 20% of the time. But it leads to this things getting lost in the shuffle. So when you're doing analyses, it's really important as much as you can to try to do ontologies and data standards. So I just really want to emphasize that point. The other thing I'm noticing is we're going to have to pay attention with how training occurs as bioinformatics evolves and develops further. We are getting into a phase now where some of the earlier stuff some of us knew back when bioinformatics first formed. Like when I was a student, there was no such thing as the term bioinformatics, but I was essentially doing that. And then the term occurred. Now we've got all these things that I think I'm finding that some students don't know about, like the transitive errors that can occur when you have a gene, say in E. coli, it's experimentally determined function. You annotate it as as gene X, I'll just say gene X, and you annotate this other gene as gene X because it's similar to this other gene at a good level of similarity. So you do a sort of, we call this annotation transfer, and then you go to Bacillus subtilis as similar to that one, and so then you call it gene X, and then you go to your uncharacterized gene and you can call it gene X, but it's actually not similar to A, so A is not necessarily equal to D in this case of these different examples, and I called this genome rot of this. We kept on having these annotation transfers that were not based on actual functional information, and so I do think we have to pay attention to that and educate people again about watching out for this, because there's a lot of information out there that you can just blast your gene and say, oh, I see that has this similarity to all these genes that have all this quote functional name, and you think it has that functional name, but actually does not at all. So cautionary note. So my second main point is that accurate, well-organized data is key, and I can't emphasize that enough. So I tended to, throughout my career, as I tell my story, this as a story, robust data, organization, having really accurate analysis methods, integration of these methods too is really what's led to some novel ideas. Okay, I'd be remiss in not mentioning this landmark of the ISMB 1998 meeting, and that is because I want to make another point. By the way, don't you love that wallpaper in the old website? This is beautiful. But basically, this is where it was so meaningful for me to get introduced, to get invited to give this talk, because 25 years ago, I gave a talk, or not gave a talk, I attended my first ever ISMB meeting here in Vancouver, in Montreal, man. And I saw my first introduction to basically Lincoln Stein talking and Terry Gasterlin. And this was important because I was like, wow, it's a girl. I mean, I hate to say it, but I was around so many guys all the time, you know, and it was like to see a woman there, you know, doing stuff and being so confident and doing cool things with databases. I was like, this is so awesome. And it made me, you know, have more confidence as a student that I could maybe go for it and I could do this. And so I really want to mention that role models are important. You know, you really do have to pay attention to this. That's why, you know, we have all this talk about, you know, diversity and inclusion. There's a reason for that, because we are going to get a lot more people's brains activated if we can just have people see that, yes, it is possible and that they can do it. Lincoln Stein was also I'll come back to that in a second, but I do want to mention that, you know, other role models for me were Joanne Dillon and Bob Hancock, because they were able to have families and have a career. And you can't tell you how many times I've been asked, you know, can you have family and a career? And I'm like, what? Yes, you can. Here's our little Brinkman lab baby boom where we had lots of babies. And I just want to say that in the lab, I mean, not me, but basically I just want to make that point though, for those that are all wondering about this. Absolutely, you can have a family and a career. In fact, my mom said to me, I always thought I wasn't around for you enough, because she is a working mom. And I looked at her like she's crazy. I never felt my mom was not around for me enough. I always found that she was there. She was there for what counted. My dad was a million miles an Air Canada flyer, and he was always there when it counted. So yes, you can do it. I absolutely, though, as my little bit of advice, I would, if it was an event that was at story time for my kids, I would say, no, thank you, I can't do that. But maybe we could move that meeting to the afternoon so that we could have it so at a good time for parents. So, you know, this is doable, but you do have to sort of prioritize your family sometimes, prioritize your career sometimes, and you can make it work. But getting back to Lincoln Stein, he was really transformative for me, because I was so enthralled with this idea of open source, open data, open science. And he brought up this data providers code of conduct. So for those who don't know about it, you can look at this paper. I'm not going to go into the details, but it's just about, you know, how to manage things like, you know, thinking of it as a contract you have with your users, and not just suddenly changing things on your website without notice, and keeping maybe old versions of things available when you change. And that led me to come up with a, quote, bioinformatics predictors code of conduct that I actually have never gotten around to publishing. I'm noticing I've got 9 of them. If I had 10, I'd get the 10 simple rules you can publish in computational biology, right? So I think I got to come up with a 10th, and then maybe you could publish this. But the point is, I've stuck it up here just for now. But the idea is that through that, you know, I was able to come up with this kind of code of conduct of what we're doing to basically, for example, you know, have things be open source, easily trainable with different data sets, and watching out for certain kinds of biases. But I have to say, for success, it's all been about people. And when we talk about sustainability in science, we have to remember the people, because this is the thing that really matters. They are the ones that keep things going. And I just put in pictures from the lab with some soccer or football photos. I don't know if I should say soccer or football to this audience, but I will say football. And but basically, sort of as somebody who has a British family and Dutch family, this last week was a bit of an iffy period. There's the Dutch-English, England and Netherlands-England match, but But I have to say, go Canada, because tomorrow they're in a third place match, just like at the Copa America. It's amazing. But okay, but anyways, I just want to say though, it's all about the people, and not just the people like in my lab, and colleagues, but some of these people working behind the scenes, like our supercomputer, the CEDAR, is an awesome resource that we've been able to take advantage of, and these sysadmins and people who've been maintaining that and helping us and supporting keeping our web applications up is so appreciated. But this material, this is like only scratching the surface about all the communities, for example. And I want to do shout out to those in bioinformatics.ca and others. There's this microbioinfo Slack that is amazing, with 2,000, at least 2,600 people in it. If you're interested, let me know. We have a policy. We don't want to release the the invite publicly, but just to people more individually we can. And but it's a really great Slack for microbial bioinformatics. But you guys will each have your own sort of communities, and these are really important to maintain. But here I want to get to some of the issues, right? Money, people cost money. I mean, this is my lab right now. These people do ching, 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 they cost a lot of money, and I don't want to pay them low amounts because these guys are awesome. And then add to it, we've got this wave of data, like insane amounts of data. I think this is more like how I feel with the wave of data right now. But I think we've got so many challenges we're dealing with. And then we've got on top of that, this, what I think is a non-sustainable grant cycle. So you get a grant, woohoo, you do develop novel software. By the way, these commentaries are inspired by Rob Biko, an awesome bioinformaticist who gives great talks if you ever want to get somebody out for a great talk. So the funding, you get this grant, you develop software, the funding ends, you apply for another grant, maybe it gets rejected, you go, but eventually you might get another grant, so yay, but that grant has to be on something a little different, a novel. So then you're ending up developing this novel software, plus you want to maintain that original software from your original grant. Then the funding, ends soon, you apply for another grant, but now this is risking both the original software and the new software. So you've got sort of software plus one. And then if you do it again, you go around, you have your software plus two, and then your software plus three, and so on. And what I think is happening is why we're facing this crunch now is because bioinformatics and computational biology have matured. They're now at the point where we have a bunch of resources that we would like to maintain, and our funding model is not set up to maintain this, and not set up to, it just basically makes you, have to get grants. So this is a paradox that was, with credit to Phil Bourne, who published a little preprint on arXiv on biological data sustainability paradox, that with the fixed funding base, the maintenance of biomedical data, and I would argue software, takes money away from innovative research. However, those data are increasingly important in achieving innovation. So we do have this paradox that we're dealing with. What's the solution? Well, thanks to my Indigenous colleagues, I really realized that I should consult first and find out more information. So I initiated a survey, which I only got ethics approval for about a week ago. So it hasn't really had very many people yet, but certainly, it's basically a survey, just one page, just a few questions. Encourage you guys. I'm going to give you a QR code to do a reply to it yourself, but you can, about sustainably maintaining bioinformatics and computational biology resources. Questions like, what is the primary way you have financially maintained resources after initial funding is ended? And I'll just give you a preview of some of the results that we got. So first, just to give you the data, these are kind of the options of questions. I thought of, my supervisor's funds, if there's a grad student, applying, employee institution, different kinds of institutions, user fees, philanthropy, society, my own money. There's actually people who replied they use their own money after the grant ends. That's sort of scary. But here's the results that we got so far. Oh no, darn it. Sorry, if I can't use a mouse. If I use a mouse, just a minute. I have to learn from you what I do at that time. What do you?
There you go.
What did you do?
Just dragged it all the way across the screen. So it's right there for you.
Okay, joy. Okay, so nope. No, it's not the mouse. It's not moving forward. What did you do last time? Sorry.
I was moving the mouse. I might have clicked.
What did you click there? Okay. Okay. Thank you. So, so basically, so these people are important. It's important to pay them too. Okay, right? Anyway, so current approach is indeed grants. So the preliminary results are that you can participate, and this is the QR code if you want to participate in this survey. The majority, are saying another grant. So what was really striking to me is how many people are actually saying, I'm getting a grant, and then if I run out of funding, I am using another grant as a way to maintain those resources. The second most common answer is my employment institution. I will put a caveat. This survey, the way it's designed, is certainly just more geared towards getting the diversity of responses. It's not particularly good for looking at enumerating this, but notice that for the additional way you want to financially maintain resources after initial funding, instead of just like a primary one, over a third said there's no other form of support. That was the most common answer, is basically they are just going for grants and getting grants, and that's the only way they're trying to sustain things. Then it was another grant or employment institution. I think it's interesting, employment institution is coming up, so there's employment institutions stepping in. So when I looked at this question about suggested solutions, lots of comments like, it's a difficult issue and there is no clear path forward. But and then the really concerning one is for issues of maintaining resources, put master students on them. I don't know. Oh, I wouldn't want to work in that lab. But more community input, less top-down decisions. That we do want to consult more. Funding opportunities dedicated to bioinformatics resource maintenance, having software data resources as infrastructure instead of limiting these funds to buying instruments only. There's a bunch of ideas. I'm going to release this later about what we find, but I want to give a credit to Phil Bourne, who really is trying to push the envelope. So he's trying to say, well, let's just look at totally different ideas. So this is just to stimulate ideas, but the idea of a cap and trade model where data and services have value in a marketplace, where the private sector is an active player and it can be driven by the economics of the marketplace. And you can see this paper for more information. But I think I just want to summarize this, though, in one sort of slide, and that is that I think the challenges are that we do want to maintain the benefits of open data. It's been really useful having data get out openly. Low middle income countries, many other, the haves and have-nots of labs can all sort of access data and do analyses. The equity for disadvantaged groups, the need to ensure data sovereignty. There's a lot of challenges that we need to factor in. So what is increasingly clear is grant-only approach to sustainability is just not sustainable, but is what most people are doing. We need, I think, more documentation of the benefits of resource maintenance. We need to really show governments that this is something that is going to really pay off if you can maintain this. And as an aside, I have this InnateDB database that was led by David Lynn, a researcher in Australia. He is still tirelessly, both of us don't have any official money to support it, but the SFU is still willing to host it, where I'm my institution, and we still reply to messages and stuff, and we still keep it going. And I was looking at this cool, if you look at Dotmatic, no, not Dotmatics, that was a mistake. There's a thing, I'll see if I can remember at the end. There is a way you can look at your papers and see what are the most influential papers that have cited your paper. And I found all these papers that have cited InnateDB that are like, high impact journal papers that have used it in the past 5 years, where we had not very much funding at all to run it. We were basically just running it on a shoestring. And think of all these things that might not have happened, if we hadn't been able to maintain that. And so I do think there's a benefit to trying to maintain resources. I do think we have to explore public-private partnerships. I do think that we do need to look at some targeted government initiatives now to mitigate the short-term serious challenges. But we have to do this really with consultation and look at how any kind of new model might cause unintended harm or unintended benefits. We got to look at, we've really got to take the time to do this and not be like, the tortoise and the hare. We shouldn't just race like the hare to coming up with some solution. We should really reflect on this and come up with something. And so more discussion and consultation is needed. So sorry if you're expecting some great answer for sustainable science, but I can say I do think we do have an answer. We do need to put some serious effort into looking into this. And we need to do it with sensitivity. So the last thing, and unfortunately I don't have a clock here, so I don't know, okay. The last thing we do need to do is we really need to look at sustainability with what I call sensitivity, but honestly, it was just an alliteration for the title. It doesn't really mean anything, but what I'm talking about is making sure whatever we're doing, watching out for unintended harm or loss of benefit to communities. This is what's really come out for me in the past year. I was always big on open science, but I've realized we have to think of it as open science with. So I'm going to do the Dutch thing of like in Holland, you basically have patat met, which is awesome stuff. You go into Amsterdam and you get these fries with, and it's understood that the with means mayonnaise. So you basically have fries with or chips with, and it's called patat met. And because the ladder of mayonnaise is understood. Well, I think we need the same kind of thing. I think we need open science with. We need open science with it's understood. We're going to have some consultation and care of looking at any impacts we might have. So now all of the things that I'm doing, I'm trying to say, okay, I've got this result. Do I need to maybe talk to this group, that maybe it's to do with COVID and the elderly? Do I need to talk to some elderly and just sort of see, how would you think if this result came out? And just make sure that maybe we're presenting this in the best way. So I highly recommend the Principles of OCAP course. It's, I should have put the site fnigc.ca, that you can take this course that is about ownership, control, access, possession of data. It's brings up a lot of concepts like how research ethics, traditional research ethics are problematic. They focus on individual consent and safety, not at communities. So we have to sort of think of the individual and communities, focusing on mitigating harm instead of generating benefits. Let's try to make sure that things we're doing are beneficial. I want to encourage you guys to really think about this as you move forward in your research. Whenever you generate any result, what kind of impact could it have? And is there anything that could cause an unintended harm, or should I consult with some groups just to see what's the best way to present something? So I'm going to give the last part of this talk, I'm going to give an example of integration of omics data with other diverse data where we think it can have some benefits and community level actions that we can take that can widely improve health of people in the environment. This is with the child study, standard for Canadian Healthy Infant Longitudinal Development Study. It's a longitudinal birth cohort that we now just call child cohort study and don't use the acronym anymore. But it is exceptionally diverse data of 3,500 kids from across Canada that are reflecting generally Canadian demographics. So maybe the income level is a little on the higher side because, of course, people who are very poor just do not have the time to go and participate in a survey like this. But essentially, this is got mental health data, pollution data, diet, home environment, how many hours a night you leave your windows open. It's like ridiculous amounts of data. It's very powerful. And I encourage those of you doing any bioinformatics where computational biology, where you are looking at some clinical data, this is a great data set that you may want to put in a request to get some data. The data includes analytes, epigenetic data, genetic data, microbiome data. And so we decided to do this exposome-wide association study for asthma. Asthma is this chronic inflammatory airway disease. It's one of the primary causes of child hospitalizations. And in fact, when we did an analysis of looking at kids in the study who disproportionately have high numbers of doctor's visits or unscheduled doctor's visits or hospital visits. All of those variables that show up are indicative of asthma. It is something that really causes, it costs our healthcare system, asthma, significantly. It is a big deal. And so we decided to, this looks messy, but I just wanted to bring up the point that there is, outcomes assessed, the bunch of different ones, definite asthma, doctor diagnosed asthma versus recurrent wheeze, looking at exploratory, XWAS analysis first that was like just, with very few covariates and a sort of more relaxed multiple testing correction, then doing a more in-depth XWAS analysis where it's like logistic regression. After looking at a logistic regression for 2,700 variables, then going and putting it down to those that were significant from round 1 with more stringent covariates and more stringent multiple testing. And apologies that you can't really see that, but I am going to show some blown up bits. But the main point is there was a bunch of significant positive and negative associations with 5-year asthma. And one of the ones I want to draw your attention to was prenatal disinfectant and cleaner use was impacting the child. And this was really surprising. And this is the benefit of putting that diverse data together, made us find something. We never thought it. We always thought of it as the child getting exposure to things, the child having things. We never thought that the mother's exposure prenatally would have an impact on the child at this level. So oven cleaner, if you use oven cleaner weekly or more, yes, there are actually people who use oven cleaner weekly or more. Definitely not in my family. I don't even know if I even used the oven cleaner yet in this oven. I don't know. But I actually think what we're investigating right now, this is new data, this is not published yet, but I think it's actually that some of these people it's their job, right? So they're cleaners and they're using this a lot. They have a notable statistically significant increase in risk of their child getting asthma if they, or recurrent wheeze if they, use this oven cleaner weekly or more. Ditto for chemical hand cleaner, like degreasing agents. And what I think is really notable to me is if they used it prenatally but not after birth, like they stopped using it from birth to 12 months or on, their kids still had an increased risk of asthma. So it's really the prenatal period that was notable, and that was really surprising to us. Daily hand sanitizer had to be daily, not monthly, or weekly, but daily hand sanitizer use was associated with asthma in the child when it was prenatal use. And so these are notable. This is notable implications. I mean, we just went through a pandemic where people were using hand sanitizer a lot. I must admit, I tended to avoid it like plague. I didn't like hand sanitizer, so it was like, I hated that going to stores. But now we have to think about the best way to communicate this information. Indigenous populations have twice the rates of asthma in Canada than the rest of the population, and they also have twice the use of cleaning products in this study. So we've been talking with them, and so far, based on my consultations, there's a lot of history there with Indigenous people feeling like the dirty little Indian. They feel like they have to be extra clean. They feel like they have to have the best lawn on the rock and be extra clean. And that is really sad. But I was really encouraged that they were saying, this is good. If we can learn this now and we can move forward positively, that's a good thing. So, but we're still, these are challenging things to bring out. How do we bring out this data in ways that is going to not cause harm? I want to also hit you with, we looked at microbiome data, didn't see big signs of changes as we started to look at mechanism. But epigenetic data, the prenatal use of chemical hand cleaner and hand sanitizer, daily hand sanitizer, is associated with demethylation at CpG sites in cord blood DNA that includes genes involved in Th2-mediated inflammation, activation, proliferation of T cells. These are things involved in our immune response, and asthma is basically a disorder of our immune response. So we're actually seeing epigenetic changes in the cord blood of the child as a result of prenatal cleaner exposure. So a cautionary notes, though, we need to iterative study these things with careful selection of variables. We found we had to watch out for some variables that just are, there can be ambiguity in them. More curation, ontologizing is needed. Consultation via the impacts of communities on these results. As an aside, ChatGPT for harmonizing data has been an interesting journey. I think it's got a long way to go yet. I really like Russ Altman at Stanford said it's like a cocky third-year medical student. I agree. I decided to ask ChatGPT to show me a picture of a cocky third-year medical student, and this is what it came up with. So not bad. So there's ChatGPT for you. But because it really confidently tells you something completely wrong, right? So I've got these few things I'm not going to tell you because I don't want it to get out. I have these few things that it very confidently tells me are wrong. And I'm going to, hold on, I want to see those improve. And I'm not going to bring them up. And I'm hoping that some of these improve over time. But I am hopeful that they will. But so to summarize though, I do want to just highlight this, in my journey as a, researcher who cared about life and got into computers and got into bioinformatics, but mostly microbiology. I did find interdisciplinary integration was really what led to innovation for me. Accurate, well-organized data is key. I think this is going to be particularly important with the whole generative AI era we're entering now. We have to have that data be good, or it's going to be garbage in, garbage out, right? And we're already seeing this, and we know that, but we really need to pay attention to that. Role models are important. It's what really brought me to where I am today, and many of you, it will help. It's all about the people, but these people cost money in bioinformatics and computational biology. We do need to think seriously about other approaches from this grant-based maintenance approach. I do think discussion and consultation is needed. We need to actually put some money towards trying to figure out this problem properly and be the tortoise, not the hare, to get to that finish line well. I do think in training, we need to train more about open science with, like this, I think we should get this kind of idea of consultation and care. And I referred to care because there's fair, findable, accessible, interoperable, and reusable. That's very a cornerstone of open data. And then with care, this is sort of, associated with this idea of collective benefit, authority to control for certain populations, responsibility and ethics. And I do think we should be using this kind of fair with care approach. Okay, in closing, I'm just going to come back full circle and say, I find it very pressing now that we need to get organized. We have this powerful generative AI that can do some awesome things if we can train it properly. We do have, as somebody who's a bit of an armchair climatologist, I'm a little alarmed, like many people, over this past year. These graphs have not looked good. We're looking at significant impacts, and climate change is going to impact our food production in the decade. And I do think we need to pay attention to working with these, this life that's all connected. And I do think we really do need to get our act together with getting organized in terms of data, because it will help, hopefully, as we start to address things like appreciating that microbes are not all germs, that maybe we have to come up with new approaches for tackling microbes, not tackling microbes. We have to come up with new approaches that work with microbes and try to work with them. We can get along, and that's a little petri dish with some microbes in it. And I think I will leave it at that, except that I will just do a huge acknowledgement to the if there's anybody expecting them to be on that list and they're not, you should be. Yes, I agree, because there's just so many people that I have to thank, because I do like to go out and talk to other people about ideas. I am the kind of person that in kindergarten I was described as works well alone or in small groups. I like to be, I don't, this kind of big thing, but you too, if you're doing cool stuff and you're smart, you can do some good stuff and you can get up on that stage and convey that information to people. It can be very useful to get some of these other ideas from other people. It's been transformative for me. I just want to say thank you again to all these people and to funders. We've got Genome Canada that has had some rocky times, but is still trying hard to come up with ways to innovate the landscape, and SFU, and having your institutional support is really important. And this child cohort study, and so many of these groups in bioinformatics in Canada have been really instrumental. So thank you so much, and I'm happy to take any questions if we have time. Thank you.
That was a fantastic talk, and I am your microphone runner, but first I want to, I'm over here, Fiona, but I have to stand behind Seth so I can read the question from our virtual audience. And the question is related to software. In your opinion, which would be a more beneficial approach, funding the upkeep of legacy software or the rebuilding of said software?
Oh, that's a good question. So, I have to say what we found in our group is that after 10 years, you got to make a rebuild that's fresh. You can't just keep, there's a great cartoon, I wish I had it here, of this guy introducing some people to a bit of software and, in a company and saying, this is our nice, cool software. And then there's this hidden monster that's underneath of all this legacy stuff. I do have to say, I think it's really important to do fresh, but that costs money and it's really hard, and this is where we have that problem. But I don't think it also depends on the particular situation, right, of how well it's been designed. But, I still have some stuff running on Pro Code, I admit. Yeah.
Just raise your hand if you have a question and I'll come running to you.
I feel like I'm in a band. I can't see you guys for the lights.
Thank you. Great talk. I have a question about the child cohort and asthma. I'm here.
There you are.
Yeah. I noticed that you had looked at smoking on your slide. Have you looked at vaping? Like if a mother vapes?
Sorry, have you looked at what?
Vaping? Vaping. Electronic cigarettes.
Oh, vaping, yes.
Yeah.
Yeah, so actually that's a very good question. I will check. I do not remember. Yeah, I don't know. I mean, certainly this cohort is still young. They're like about age 13 now. We're just making the survey for that now. I'm pretty darn sure vaping would be on there, but yeah, it's a very good question, actually. I'll have to look at that for the parents, at least when their kids were young. Thank you.
Great talk, Fiona. Fantastic. Thank you very much. My question is, again, for the same study and the use of confounding factors. So, for example, people who use oven cleaners every day would be professional cleaners, would be lower social economic and status and so on. Hand sanitizer actually is interesting because that would be maybe a lot of hospital workers, for example, that would be using it, incredibly frequently. That would be my thought, and there might be a difference there between, say, physicians and others. So, have you checked for those kind of confounding factors rather than the pretty plausible mechanism that you've shown?
Yeah, no, absolutely. So this is the biggest issue, right? Correlation does not imply causation. So, I can tell you we're already just like literally this is new data. We're still just looking into this and welcome these thoughts. But yes, indeed, these people who are frequent hand sanitizer users are disproportionately in the healthcare system. Interestingly, they are not disproportionately doctors or nurses, but the administrative staff working in a hospital are also using the daily hand sanitizer more. So it's just being in a hospital environment. This is all pre-COVID, this data, just so you know. But there's a lot of issues regarding, like, for example, asthma being negatively associated with timing of food introduction. I think that's just because these are people who, if they have a family history of allergies, they're delaying their food introduction of peanuts or whatever, but they have this atopy or sort of allergic phenotype that might be more like asthma, but they are also delaying their food introduction. So you have to be very careful with this. I think that's the surprising thing to us about the cleaners is that this is correcting for family asthma history, is collecting for antibiotics courses and other things that are known to be associated with asthma. Having an older sibling is really protective actually from developing asthma, and having a dog is also. And I think it's literally you get out in the environment, there's this hygiene hypothesis of just getting those good microbes, and getting exposure to good microbes is good. I took my kids and we dipped them in Mexican water to give them like microbes. It wasn't, my daughter got a little sick after one of the incidents, but it was, it just, they don't have asthma. They don't have any allergies. But we did, but I will add that just in terms of covariates, we did correct for household income, a bunch of other things, delivery mode, and we're still seeing that, and it's statistically significant. So I think it's worth investigating more, let's just say. Yeah.
Hi, Fiona, wonderful speech. Thank you so much. I really liked what you said about community collaboration and consultation. My question is sort of related to the fact that, from my experience talking to some local community members in my community, that they're a little skeptical of people in research and academia because they feel like these are people in ivory towers who just come to them when they need something, and they don't really have the community's interests in heart. Do you have any advice for doing consultation or community work in a way that's mutually beneficial?
Yeah, thank you for bringing that up, because that is so important. I think it's really important that two things I'm going to bring up. If anybody's interested in doing some consultation, you have to take time with this. This is not something where you can just go, hey, come up to them and just ask for some input. It's about relationship building, and it takes time. And that's the sort of approach I've been using. It's about really forming a relationship with these groups that you're wanting to talk with and get input from, and making sure that it's about, hearing from them and what their interests are, and co-creating, ideas where it's, they are in the driver's seat of, this is the issue, this is the thing that we need, this is what will confer a benefit to us. I think that's really, really important right now. And I welcome thoughts. Maybe after we can chat too if you have any other thoughts on this. But I think the biggest thing I've learned is, and there's others too, like Wyeth Wasserman, who's been doing some great work in this area, that, we just have to listen and take time. This is really important. I've learned some really useful things about how to proceed that I think could really help avoid some of the situations we've had in the past. Like in Canada, we have this common CV system. It's anyways, don't get me started on that. But let's just say that was because it was made in this way that was, if we'd only got Indigenous populations involved and some First Nations involved in developing that, because we would have had this consultation and development of it, and it would have been made probably in a better way in the end, and saved a lot of time and money in the end.
And from the online audience, since they do not have the benefit of getting to talk to you after this session at the welcome reception. And the question from Viva is, thank you so much for a great talk. And this goes back to your study with the hand sanitizers. How is bias due to the pandemic use of Purell and others type of hand sanitizers accounted for in your analysis?
Yeah, so these data are from pre-pandemic. So this is not, no, it's a great question. And I think that's one thing I've got to really emphasize when bringing these data up. This is a cohort that was mostly born in 2005 or so. So this is all pre-pandemic, and I will add to just the question that's very common that I get is, so are we going to see then a big, increase in asthma, or have we seen anything? So the thing is, they're not actually old enough yet. So the kids that were prenatally exposed to cleaning products may be a little bit more than normal, maybe, because there's many people who just hunkered down and didn't actually use hand sanitizer like crazy. But for those who did, those kids aren't yet of the age that they would get doctor-diagnosed asthma, so we don't really have that data yet. But I'm not actually, from what I've understood, is there's a proportion of a population that was using hand sanitizer a lot, but there's certainly lots of people who were just, not, certainly not using it massively unless they had to go to a store or something. And it was for a relatively short period. But I do think it's something that we need to look into more, for sure. So thank you.